everyone, thanks for coming along. There's a bit more sun that side. We've had two great talks if you're here listening, or later on maybe you'll be watching us or listening to us from elsewhere. And so we've heard all about the wildlife that we might have in our backyards. From Stephanie we've heard about dragons, and from Lisa we've heard about a variety of different wildlife species. So I'm going to talk a bit more about wildlife in our backyards, but a different way that we actually can cause an impact to wildlife. So for the first part of my talk, I probably won't mention wildlife too much at all. We're actually going to talk about bacteria and disease, okay? So unfortunately, humans are renowned for polluting the environment with plastics and pollutants of various sorts, but we also pollute the environment with our microbes. And now we're seeing that our microbes or disease-causing organisms are making their way into wildlife and potentially impacting their health. And so I'm going to put some background information to the work we're doing in understanding that issue, and then talking about the citizen science project that we have called Scoop a Poop and how you can get involved in this program. Alright, so we need to do a little bit of study or background information on infectious diseases and understanding disease movement in the environment. So infectious diseases are illnesses caused by bacteria, viruses, fungi or different parasites. And pretty much we all know we get a cough, we're not a flu, that's a virus, we get a cough or a cold or a bacterial infection, that often we would go to the doctor and we would receive a treatment of antibiotics to eradicate that disease. Okay, so the next thing, what is an antibiotic? So an antibiotic is a drug that's widely used in human medicine, wildlife medicine and domestic animal medicine, but also for other reasons, for prophylaxis in production animals and so forth. And so basically an antibiotic is a chemical that actually kills or inactivates the bacteria. So when we take an antibiotic, it's typically targeting the bacteria that's actually causing that disease or infection in us. So we've got a bit of a global problem at the moment, is that bacteria obviously want to survive, so our antibiotics no longer work. And this is a global issue for now human health, but it's also now a global issue for animal health, or becoming more of an issue for animal health. And so what's happening is the different antibiotics that we have to treat our diseases no longer work. Bacteria are fought back, and now they're able to just happily survive when they're exposed to antibiotics. So basically, no effect. Here we've got some antibiotics and some, it hasn't got a pointer, usually I have a laser pointer. So, and some different bacteria sort of within reach of the antibiotics, so there's really no longer an effect by these. So how did this come about, okay? How did bacteria become resistant to antibiotics? Well, antibiotics and bacteria have actually been acquainted for many millions of years. It was just that humans discovered the power of antibiotics to treat disease that we actually started to harness this fantastic ability that bacteria had to use antibiotics and produce them. So bacteria actually produce antibiotics for signalling and for communicating to other bacteria. I've mentioned that antibiotics kill bacteria, so they also produce antibiotics to protect the space around them. So you can imagine down in the soil here, we've got millions of little bacteria, different species competing for the same resources and same space. So if they're producing a little chemical that's going to keep the other bacteria away so they can dominate that space, it's beneficial for them. And so bacteria produce a lot of different antibiotics, but early in late 1930s, Alexander Fleming discovered the first antibiotic, which is penicillin. And that name's probably very common to you all, because I think most of us would have had penicillin or a similar antibiotic in our lifetime. Interestingly, an Australian, Howard Florey, a chemist from South Australia, was also involved in the discovery and getting penicillin out and onto the market to treat disease. So what happened was, in that environment, bacteria in this little soil community, say underneath us, who were being exposed to antibiotic, well, they wanted to survive, so they ended up evolving or acquiring superpowers, and so they can actually resist the antibiotics that others around them were secreting. So when humans came along and started using antibiotics, producing large amounts of chemicals in production facilities and polluting the environment with antibiotics, we started selecting for those the ones with superpowers. And so eventually, when in humans in hospitals, we started to see the emergence of antibiotic resistance to different bacterial diseases. And then eventually now we're seeing those bacteria widespread, not just in hospital situations where people were compromised, but widespread in the environment. And occasionally we'll have someone who's a very healthy individual, they might cut themselves in the backyard and acquire one of these superbugs, and within 24 to 48 hours, they've unfortunately passed away. So we do have some rather extreme outcomes now of these what we would call multi-drug resistant bacteria, and so they're different to lots of different antibiotics or antibiotic classes. And basically, to kind of explain it, here's our, keep going for the laser pointer, here's our bacteria that's acquired antibiotic resistance. It's like having an umbrella. When we pour the antibiotic on top of it, it just bounces away and they're able to continue growing and the disease continues in that particular person or infected animal. All right, so the other component that's kind of happened is that the biological traits that make the bacteria resistant to antibiotics are actually pretty unique, and they're mobile, okay? So they can actually move between different bacterial species or individuals within a bacterial species or across species. And this is one of the reasons why we've had this rapid emergence of antibiotic resistance around the world. So it's basically like if I've got the antibiotic resistant gene or specific trait for penicillin, and I bump into this bacteria over here and just pass my little package along, we're both now resistant to antibiotics, okay? And so what we're now seeing is the friendly bacteria that live inside us, things like E. coli that's in all our guts and quite common across different animal species, including us, now have acquired the ability to resist antibiotics. So the friendly bacteria now can cause disease, okay, or some of the friendly bacteria, not all of them. We still have friendly types of, say, E. coli, but we have more of the types of E. coli that can actually cause disease. And the same for other ones. And so the one you might be really familiar with is golden staph or MRSA, okay? And a lot of these organisms are actually just out in the environment. They don't always have to be involved with what we would call a host or whatever they're infecting. All right, so Australia's responding to antibiotic resistance through the National Antimicrobial Resistance Strategy, and it's within a framework we call the One Health Framework, okay? And basically what we're saying is we're all connected to our ecosystems, we're connected to our wildlife in our backyards, we're connected by water, we're connected to, our wildlife are connected to us by inhabiting our backyards, by kind of feeding in our bins and on our waste, and then the environment. And so basically whatever happens here, here and here flows on and can impact on the other compartments. And so to understand what's happening in humans with antibiotic resistance, we need to understand what's happening in what's happening in the environment, and then also what's happening in wildlife and domestic animals, because everything's connected. And that's how we need to tackle this global issue, which is how it's being tackled. Okay, so now we'll get to the animals. Okay, so we're trying to understand, basically antibiotic resistance now has no boundaries. As I said, it emerged as really a primary human issue in hospital patients. It's now spread outside of hospitals into the community, and we're now seeing it in our wildlife. So we've been investigating antibiotic resistant bacteria in wildlife for a number of years. We're looking at movement of these resistant bacteria into marine systems, looking at species in urban environments, so flying foxes and possums, and then also thinking about what happens in conservation. So when we're managing threatened individuals, threatened species in captivity, breeding them and then releasing them to the wild, and understanding the risks of resistant bacteria there. Okay, so how do we do this? It's very easy. Well, sounds easy. We start out with collecting a fecal sample, and so here, if you've got possums in your yard, you might recognise this particular fecal sample. So this is a brush-tailed possum sample, and then this one here is actually a very chewy, sticky. I shouldn't say chewy, should I? It looks chewy. It very much seems chewy and looks like a piece of caramel when you collect it. But this is us collecting a flying fox fecal sample out in the wild. So we'll grab that fecal sample, we'll take it into the lab, and we'll actually extract the DNA from that sample. And then we do, or we'll grow the bacteria, and then we'll do a test to work out if resistance genes are present, and then we can actually look at the little code in the DNA and we can actually say this bacteria is resistant to penicillin or erythromycin or whichever antibiotic we're interested in or they're resistant to. So we apply that, or we've been applying that to different wildlife species, and this is just to show you what happens with some of our data across those species and the extent of the problem. In the pink, we've got captive animals, okay? So we would assume that if wildlife species were going to acquire resistance, the place that would most likely happen would be in captivity, where they have that greater contact with humans and our environment and the pollution that of those bacteria that we put out there. So that's exactly what happens. We can see that in sea lions, little penguins, possums, bats, rock wallabies and echidnas across the board in captivity, they all have quite high levels of antibiotic resistant bacteria. Okay, you can see the numbers we tested up here, and that, you know, some species are sitting around 50%. In contrast, you might look at, would then predict that in the wild, perhaps populations of that species that are far removed from humans may not have as high levels of what we see in captivity. And we did see that in sea lions, and so here we saw a 0 occurrence, but then you can also see that we are seeing resistant bacteria in some wildlife species. So very low in the little penguins, but at least around 2%. In brush-tailed possums, close to 30%, flying foxes just under 15%, none in the wild for the brush-tailed rock wallabies, and then out here the echidnas. This is a new study, so the numbers are quite low, sitting around 23%. So what's that telling? That's telling us that, or it's suggesting that, this is the kind of questions that we're asking at the moment, that, you know, is there a greater occurrence of antibiotic-resistant bacteria in urban environments versus rural environments, and thinking about how these bacteria and what types of bacteria are moving into different wildlife species in those different ecosystems. All right, so what's the significance of this? Okay, so to humans, resistant bacteria can cause life-threatening disease. Equally, if these bacteria make their way into wildlife, there's the possibility, which they are, there's a possibility that they're disease-causing organisms, so we may actually inadvertently spread a disease and see a new disease emerging in a wildlife species. If we've got background antibiotic-resistant bacteria in our wildlife species out in the wild, and we've just heard from Lisa that we have a number of animals coming into care, often they will need antibiotics, okay, because they do have an injury or they have an infection. That antibiotic may not work because the bacteria that they've actually got in that infection is resistant to antibiotics. So we have implications for treatment when they come into care. We have to remember that these bacteria are really invaders. They're like weeds. They shouldn't be in those particular wildlife species, so they may be impacting their health in some way, or disturbing the bacteria that should be there, and so that's another potential risk. And then if they acquire resistance when they come into care, there's the potential that those bacteria may be released back into, or if they're in care and they've come in with resistant bacteria and that relationship is changed some way, when they're released, they may actually take those bacteria to populations and again increase a risk of disease. And then also, how far have antibiotic resistance bacteria spread in wildlife? And so these are all questions we don't know, and that we're really trying to address. And the big one for us is, how far are these genes spreading? And one of the complications with that is we do a lot of field work, to collect our samples, but to look at this question we need to look across a great expanse of land, and we can't do that alone, and we just would never get the funds to sample all of Australia, and that's where our citizen science project comes in. So we have a citizen science project called, and you've heard the name, Scoop a Poop. This is our app page, and basically citizens tackle resistance in the wild. So what we're doing, we're going out to schools or community groups, we're giving a talk like today. I go into a bit more detail about the issues of antibiotics. We provide people with some training on how to collect samples and how to recognise the type of sample that we're after. We pass out a Scoop a Poop kit, and I have one here, like this. Which way does it go? We have a Scoop a Poop kit, and then people take the Scoop a Poop kit home and they collect a possum sample from their backyard for us. We probably don't have people from rural areas here, but we're also reaching out to rural areas, and if we can get we can get possums, we can, but we've also started doing some work on kangaroos and other species as well, just to also get people involved. So we've got our kit, we've got our citizens who collect the sample, we do a lot of work with schools, and then what we're actually getting is this map of poo of Australia and a map of results at the same time. So our citizens can go in and can actually also track the results of their sample. And yellow means it's still being analysed, red means it was positive for antibiotic resistance genes, and green means it was clear. And if you hone in on the site, on the map, you'll actually see we've got hundreds more samples around Sydney, and that we're actually now just starting to expand into other states. And so basically we need help to ensure that we can cover the range and get a sufficient sample size for our study. All right, so if you want more information, I'm around for a couple of hours. We also have a tent just around the corner. Our collaborator here, Koa, I think it's tent number 16, and you can come up and have a play with a kit. Oh, we have
a possum on our
not a real possum. We have a possum. Possum.
And we're next to the powerful owl, so you'll.
There you go. So and if not, you can follow us on social media as well and see what's happening.
Thank you so much, Dr. Michelle.